Good afternoon, everyone. Welcome to the sixth webinar of the Quality Webinar Series, jointly organized by EATRIS and ECRIN. My name is Laurent Duquesne. I am Education and Training Manager at EATRIS, and I will be hosting this webinar today. Today, we are very pleased to have two speakers joining this webinar about quality and reproducibility issues related to clinical development, Christine Toneatti from ECRIN and Raphaël Porcher from Université de Paris. Before I introduce our speakers, I want to make you aware that this webinar is being recorded, including the questions and answers at the end of the session. It will be made available on the EATRIS website shortly after this session. We have reserved some time at the end of the session for questions. We invite you to write down your questions during the session in the question box in the control panel, as you can see on this slide. The first speaker today is Christine Toneatti. Christine is the Head of Quality and IS Unit at ECRIN's Central Paris office, overseeing the quality system, managing risks, and optimizing access and security to ECRIN information systems. She has over 30 years of experience in biomedical research, initially as a researcher and project manager in the field of infectious diseases, including involvement in drug and vaccine development programs, ranging from research and non-clinical development to clinical phases 1 to 4, and epidemiology. Since the year 2000, she has provided leadership in the development and implementation of quality management systems in good cross-practice regulatory, research and ISO environment for various industrial companies and academic organizations. As an international auditor, she has coordinated and performed non-clinical, clinical and information systems audits and hosted inspections for governmental agencies in Europe and Japan. She has an MSc in parasitology and another MSc in quality and research management, awarded by Cranfield University in United Kingdom. She is ISO 27001 2013 certified and has degrees in statistics and ethics. The second speaker today is Raphaël Porcher. Raphaël is Associate Professor of Biostatistics at Paris Descartes University, Université de Paris. He is also co-director of the Centre Virchow-Villermé Paris-Berlin and a member of the methods team of CRES UMR 1153. As a biostatistician, he is involved both in methodological and statistical research, as well as in applied clinical research, including clinical trials, observational studies, and prognostic studies. I will now hand over to Christine. Christine, it seems I cannot unmute you. Could you please try yourself?
Yeah, is that okay?
Great. Yes.
Okay, thank you, Laura.
Thank you.
Thank you, and good afternoon, everybody. So I will go to my presentation, and just to set the scene, what I would wish to cover this afternoon is really the arguments and rationale behind the need for data quality and for data sharing, before to touch on some of the issues and hurdles that are recurrently raised regarding data management, especially by our authorities. I will also briefly summarize the rules that are applying to clinical trial data and principles for data sharing. And then I will cover, as an example, explain the program that we set up at ECRIN to implement and enhance high quality data in academic data management units before to end with some perspective and hand over then to Raphaël. So just to set the scene, let's say, in recent years, I would like to say that a big cultural change happened in the community and researcher community with the handling of research data that has resulted in the promotion of a culture of openness and an increased sharing of data. This shift in the mindset of the community for sharing research data is linked to the fact that advances in all areas of science depends on free movements of data when maintaining personal data protection. And I put on my slide here some of the prominent publication that highlight this point and advantages of sharing clinical trial data. Currently, data sharing is perceived as part of human rights and, in fact, providing true potential benefits to the patients by pointing to new research questions or giving the ability to include results in meta-analysis and also giving confirmation of published results. Some initiative already have been already set up to promote the sharing of clinical trial data and also to enhance the transparency of research results. I put some in the slide here. You might know that following publication of the Lancet series on waste in research, the lead authors, Chalmers, Ioannidis, Glasiou, set up a website that is dedicated to the sharing of information and promoting resources regarding how to increase the value of basic and applied research. I'm speaking of the reward website. It has been estimated that 85% of research is wasted, usually because it asks the wrong question or is badly designed or because the quality of the data cannot be trusted. So this is a true concern. And I mentioned here also on my slide the recent analysis published by Christian Oman in 2018, classifying data sharing processes in 9 main processes and sub-processes that highlight that data sharing shall be planned before data collection from the very start of the clinical trial, if we are speaking of a clinical trial. It shall obviously embed in the picture some specific tools for the identification of the data, and it implies compliance to regulatory requirements and consent of the participants. All this works signing that this cannot be performed without designing the trial from the very start to embed robust, predefined rules for data management, and this throughout the whole data lifecycle. Moving now to some issues and hurdles that are recurrently raised by our authorities. I would like to draw your attention on this slide, which is summarizing the typical challenges noted by the regulatory agencies over the past 50 years. And if you look at this, you can see that non-contemporaneous recording was one of the prominent finding and observation, with document backdating, copy of existing data as being new information. In labs, rerunning of samples to obtain better results, and at the end, data fabrication and discarding of the original raw data. All of this was obviously raised in the context of a paper acquisition of research data. Now, moving to what they are raising recurrently over the past 5 years, in a totally different environment, because more and more e-systems, electronic systems are used to, at least in clinical trials, to acquire the research data. You can see then again, data fabrication is there. Here, much more in link with the metadata manipulation. Still missing data and data removed from the metadata sets submitted to the authorities, duplicated data. The lack of user requirement specifications, and here we are touching on the e-systems new environment. The lack of review of the audit trails that are not configured in the dynamic format, which is very important to keep in mind, because this means that it's very difficult also to be able to reconstruct the research and clinical trial history. The risk-based software validation that I mentioned is a constant finding for the during the inspections of the authorities, because if it is performed at the very start, it's pretty much the case that this is not anymore re-evaluated on a continuous basis and throughout the entire GCP data lifecycle as it is required. More and more, as you know, subcontractors are part of the data management organization for one specific trial, and more and more we are using cloud computing. So it's obviously difficult to understand and stay aware of where are your data, where are located your data, and especially to ensure that they stay located in Europe as per GDPR regulation requirements. And overall, there is a lack of oversight of sponsors on e-vendors, with a lack of contract, provision of staff training, lack of audit, weak communication flow, and lack of traceability of data and data flows. These are under the loop of the authorities that are focusing on this lack of data quality and reliability over time. And I put on my screen here the EMA GCP inspection outcome. This is an extract of the annual report of the GCP inspectors and GCP question and answer report. And as you can see, the essential documents, source documentation, and data management are still placed among the highest category of a finding, either for the routine inspection and for the triggered inspection by the EMA. Moving now to another part of the world, and when looking at this, the analysis of Barbara Unger that has been recently published, she analyzed all the warnings letters from the FDA citing data integrity failure. And as you can see, there is a kind of peak in 2017, in 2018 regarding all findings related to data integrity, with a lack of mapping of the data and process flow. The system can be validated, but not necessarily for their intended purpose, which is the requirements. And and there is a lack of data governance process of the sponsors regarding the subcontracted activities. This conducted obviously to enforcement actions for the FDA, delays in the registration of products, and this is for the EMA and for the FDA, re-inspections of some sites, and at the end, it can requalify the ability to register the drug, so possibly, obviously, impacting the availability of drugs for the patients. All of this are related to human, technical, organizational causes, and as it is summarized on my slide now, it's performed nowadays in a very complex environment for the data management and data handling for different reasons, because we have multiple data sources with medical records, of course, lab reports, of course, but also a lot of central data acquisition or review or reading, which are much more involved in the nowadays clinical trial design. We have also multiple information type with raw data, analyzed data, metadata, images, and now and for tomorrow, mobile device data that could come from ePro device or wearables. We have also multiple storage systems. It could be local hard drive, shared drive, cloud computing, SharePoint, central or subcontracted data centers. And putting all together with the increased interface and subcontracting, and when looking at the process mapping for the data flow, you can obviously understand that at the end of the chain of custody, it's quite difficult to understand and to identify the data owner. It's very difficult to guarantee an appropriate level of access right control, and infrastructure and databases are more and more complex and externalized, which is making the whole journey very difficult at the end for the sponsor. Speaking of the rules applying to data quality during clinical trial, mainly the ICH GCP E6 R2 is the reference and is under revision. We have had also strengthened rules with an increased set of guidelines and regulation that have been published to establish data integrity standards. And I just put here in 2003, the US 21 CFR Part 11 regarding e-records and e-signature, plus at the very end of my slide and list, the EMA guideline on the TMF content and management. Just to bear in mind that whatever is the media on which you acquire your data, paper and or electronic or more and more hybrid system, the rules are the same for the regulators and shall be applied to ensure the same data reliability. And this recurrent observations related to data management and document management led to a context of increased controls and sanctions regarding the data management in clinical trials, with an enhanced inspection, more and more requests for evidencing the risk-based computerized system validation, the requests for having business continuity and disaster recovery plan in place, a reinforced set of control for the access rights, the control also of evidence for personal data protection, and I spoke about the data location also, which is one of the recurrent issue and lack of awareness that is raised for at the sponsor inspection. And at the end, the assurance of a risk management and evidence of data integrity and security for the whole data lifecycle. Why? Because JXP activities generate informations. This is the output of what we are doing. And these information are obviously used to take health decisions. So this is deemed as one of the very important measure that shall be assured during clinical trials. Regarding quickly the requirements for GCP compliant data during clinical trials, this is quite simple to say, more difficult to achieve. Data shall fulfill the ALCOA principles that are listed here, means they shall be attributable, legible, contemporaneous, original, accurate, complete, consistent, enduring, and available. I'm insisting on this two last because it means they are under the loop of the authorities, and it means that the media on which the data are recorded shall be controlled and by period assessed over time for the whole duration of the retention period. They shall provide any reader or inspector or auditor with the ability to reconstruct the clinical trial. And when speaking of available for the lifetime of the record and retention period, this means 25 years according to the clinical trial regulation, whichever type of supporting media is used, paper or electronic, which is obviously very challenging to achieve for some of the trial that we are speaking of. Now moving to what are the principles that are currently set for the reuse and sharing of data after the clinical trials. Or the FAIR principle have been defined as very important to comply with, which stands for findable data, accessible, interoperable, very important, and reusable. And again, it's easy to say, but difficult in practice to achieve. And I would say that you can here have a look on the classification of processes that is proposed by Oman and collaborators for sharing individual participant data. This is the very start of that work, and still we have room for improvement and setting up practical tools and practical policies and a full framework to get the ability to achieve this sharing of clinical trial data. Now coming to the implementation, how do you engage with your data integrity journey? Obviously, as you might have understood from what I stated before, this means that you have technical controls in place, operational and organizational control in place too, to ensure the information security and data integrity. I put here a set of 3 slides where are summarized the technical controls that have to be put in place. This is really just an overview, and as you can see, technical controls, you shall start with some requirements that are very basic, but that you have to achieve, related to the database security and maintenance, the data protection, authentication. We spoke about the access rights and the control regarding the access rights. Also the testing, backup, restore and availability of a help desk to end users, And I spoke already about the business continuity plan to be able to get back to paper if it is needed, and the disaster recovery plan. Speaking of the operational controls, just to sum up, one very important thing is really to set up a data management plan that is mapping the data flows. And why is it so important nowadays? Because the data flows are, as I said previously, quite often splitted in several organizations, some that are acquiring the data, others that are dedicated to, as subcontractors, manage the data, others that are possibly involved in providing the tool for managing the data when we can think about orders at the end of the chain of custody that will be involved in archiving the data. So a lot of third parties involved. So it's very important to figure from the very start how it will be, how the sponsor will get the oversight of this whole life cycle. Avoiding non-conformities and assessing the changes. For the same reason, it's very important in the clinical trial environment to be able to be prepared to the change, because it's quite often the case that our protocol have to be amended, as you all know, during the course of a study. So how will it be managed, especially considering the whole and complicated, complex chain of actors that I mentioned? How will it be managed and ensure that the change are well reflected in the data management? This is very important. It's obviously important also to minimize the human data processing, because it is a cause of risk and errors, and to embed automatic checks in your data processing. And behind this procedure and quality control shall be enforced. And but this is not always easy to achieve. Minimizing risk in data management means minimizing data transfer, but as I said, obviously we have more and more that kind of transfer, so it shall be from the very start secured with data encryption and security during transfer. Organizational control, this is all related to human and process. So I'm speaking here of good documentation for filing and archiving the data integrity training, not only at the sponsor level, but for all involved third party, escalation of non-conformities and the ability to put some corrective and preventive action, the risk-based computerized system validation for all the systems in scope of the clinical trial data acquisition, not only the EDC system, but all the systems. The ability to independently have self-assessment, regular self-assessment or independent audit of the performance, and obviously to maintain the oversight on the IT vendors through robust agreements. ECRIN put in place, integrated this set of control in a program that is called ECRIN Data Center Certification Program. We embedded all of these practice and controls in a set of informatics and data management standards. Taken together, this would describe GCP compliant IT and data management data, with the emphasis on pragmatic measures that are feasible for non-commercial units, academic units. These standards could be used as a standalone set of reference documents, and they are freely available, and that they were intended to be used for quality management, but for validation, for the preparation of inspection or audit, and obviously also for staff training. And we associated to that robust process of audits, data integrity audits that are performed at the data management units that voluntarily apply for certification, and these audits are confirming their ability to provide compliant, effective, and efficient data management services.
Christine, you have 3 minutes left.
Thank you. So briefly, the model components. So I spoke of the standards. We have 14 qualified auditors and an independent certification board that is assessing results of the audits to decide for certification. The standards themselves are based on the regulatory regulatory requirements and on the guidelines. We are regularly reassessing these standards, and this is altogether 106 standards that are split in 16 lists, covering general standards for training, IT standards, and data management standards, and some specifics on treatment allocation. Quickly speaking of the procedure for being certified, this is going through an application and a voluntary application of the centers. Then this application is assessed for readiness by a certification board, and the audits then take place with an independent assessment of the results that lead or not to certification of the size certification that is maintained over time. Up to now, we have 13 certified centers in Europe and some more in the process, but we conducted 41 independent audits up to now. And I would like to briefly touch before to end on the fact that as being of interest for Eastern countries, we initiated a collaboration in 2017 with some of these Eastern countries, which led to expanding the program in Japan and more recently in Korea, with the first audit performed at Seoul data center in 2020. So this data center certification program is really here to ensure the quality of the data management when performed in academic non-commercial unit. It is rewarding also through the sharing of latest technical development and training of auditors. And this is promoting overall the harmonization and the standardization of data management practice. Some perspective before to end my presentation, just to say that this implementation that I spoke about is also set up through various other initiatives. I'm speaking here of the BSI initiative for accrediting the investigator research sites. Also, the eClinical Forum put in place some self-assessment readiness tool for investigator sites. In the UK, you have a registration process that is in place for quite a number of years for units involved in clinical trial to ensure and certify that they are producing high quality data. So so this is really something that is under the loop that we shall testify and give trust in the data that are produced in clinical trial. For clinical data sharing, as you might have understood, there are some room for improvement still in front of us. Especially, we need some tool. We need to have some more training in place, and to improve the platform and repositories that are in place or currently developed, before to be ready to say that we can start really with data sharing process that are enough robust in the near future. In conclusion, I would say that education is needed in data integrity. We shall provide data trust and credibility. This is essential in light of what I mentioned regarding the recurrent findings from the authorities for the data management. And it is essential also to implement robust quality system throughout the whole data life cycle, and to start to anticipate how we will share the data tomorrow. I put some list of reference in various sectors that I covered for you to have a look at this if you would like to learn more about this. And now it's time maybe for some question. Instead, Lohan, if we are late and if I have to hand over maybe to Raphaël.
We usually have all the questions at the end for both Raphaël and you. So you can hand over to Raphaël.
Okay.
Thank you, Christine.
That's done.
Thank you, Christine. So it's up to me. I will speak of something completely different. Do you hear me? Yeah. I will speak about how to validate or what should we want when we talk about a biomarker for precision medicine. Am I able to I don't know how to. Okay, like that. So a biomarker is generally defined as a molecular alteration that is measurable and that defines a biological phenotype that characterizes a physiological or a pathological state. There are many works on what do we measure, where, in which type of tissue, how, and that's absolutely not my topic here. And I can even consider anything as a biomarker. Age can be a biomarker, imaging, whatever you want. And there are three. Do you see my screen or not? No. How can I? Okay, let's.
No.
If
now
I
we
put
see
it
your
in
slides.
screen, you don't. And let's. No. Let's try something. No, I can't. Okay. If I do that, then you will see the full screen. It's better. Good. Sorry. So there are three key properties for a biomarker that have been proposed for genetic biomarkers, but that can be extended to any type of biomarker. The analytic validity is the ability of a genetic test to accurately and reliably measure the genotype or analyte of interest. The clinical validity is the ability to accurately and reliably predict the clinically defined disorder or phenotype of interest, and the clinical utility relates to the evidence of an improved measurable clinical outcome when we use the biomarker, and the usefulness and added value to patient management. And clearly today I will only tackle the last issue, and I'm not competent at all for the others. Sorry. So for precision medicine, we focus on clinical utility, and clinical utility has been shown not to be so high or not so well defined or evaluated in practice. And and there are many works. So I cited two of PhD students of mine, former PhD students of mine, but there are many others that show that often more emphasis has been put to the other aspects of biomarkers. And one reason is that a lot of trials using biomarkers are enriched on the biomarker, and we will see later on that it is not a good idea if one wants to evaluate the clinical utility. Moreover, we may discuss what do we want a biomarker for. There are biomarkers for diagnosing a disease. There are biomarkers to refine the prognosis of the disease. There are biomarkers to monitor the response of to a drug. There are biomarkers to predict the response to a drug. And for precision medicine, again, what we want is response prediction. So we want markers that are associated with the response to a treatment in terms of efficacy or toxicity, and not merely prognosis. So what's the difference? Here, if I can, I have lost this thing. Sorry for that. I don't know how to have it on the same screen. Okay, here is it. Sorry. Here is it. I hope it will work. If we look at the sorry, I'm not very proficient with that. Okay, and now I can try. So if we look at these survival curves, these are taken from an example of metastatic breast cancer patients. Some received trastuzumab plus docetaxel, and other one had pertuzumab on top of that. The first group had placebo instead. And we have divided the curves by a marker, which is PIK3CA white type or mutated. And what we want on the curve, what we see on the curves, is that the placebo curve, which are the blue ones, are always under the yellow one, whether they are dashed or dotted, which are the white type patients, or plain, which are the mutated patients. So in each case, so clearly the marker is prognostic. That when you are mutated, you have a worse progression-free survival. Sorry, but in whether you have that worse survival or not, if you give pertuzumab on top of trastuzumab plus docetaxel, you have a better PFS. So this is only a prognostic biomarker, and it doesn't help me to decide who to treat. Conversely, if we look at those curves, which are from advanced non-small cell lung cancer, patients were treated by erlotinib in yellow or placebo in blue. On the left, you have EGFR mutated patients. On the right, you have EGFR wild type patients. And we see that the yellow curve is much above the blue one for PFS on the left. So here. So for EGFR mutated patients, erlotinib does better than placebo, whereas it does more or less the same for EGFR wild types patients. So in that case, we have a predictive biomarker that can be used for treatment selection. Now I have to remove those. Sorry. Can we remove? Here's it. So we will take the example of a fictive example that I have borrowed from Holly James, who has done very interesting papers on the topic. And this is the example of estrogen positive, node positive breast cancer, but it's a fictive example. And the outcome would be 5-year disease-free survival. Then we have two groups that would be tamoxifen, treatment by tamoxifen alone, or tamoxifen plus chemotherapy. And what we know is that on average, we obtain 5 years DFS of 79% with chemotherapy and 76% without. So not a big difference. And we will try to look at fictive biomarkers to see how we can find a good biomarker. So first, we can have here two biomarkers, the A and the D. And both, and D is even a stronger prognostic biomarker, which means that when you have low biomarker values on the left of the curves, you have a much worse DFS rate than when you have high values, sorry for that, of the biomarker. But in each case, the group who received chemotherapy, the dashed line, is above the plain line, so the biomarker can be strongly prognostic, but it's not useful for treatment selection, because in each case, we would recommend chemotherapy for all those women. So we can also ask, what can we do with a single-arm trial? So in a single-arm trial, we can have such a curve for a new marker B. We have a better DFS overall, it's 84%. And we might say, well, 84% is better than 79% for those on the left. So it's good to decide treatment. But in fact, we don't know how those patients would fare if they wouldn't be given chemotherapy, and perhaps there would be the same curve, but just under the other one, or over the other one, sorry, or under. And so again, it's not useful for treatment selection. We could also ask ourselves what we get in an enrichment design, and there are many personalized medicine trials that use enrichment. So only patients, biomarker positives, can be recruited in the trial. An example would be a BRAF mutation for dabrafenib, which is understandable, in fact. But so if the trial results are favorable, it does not imply that a marker should be used for treatment selection, because we don't know what would occur to the biomarker negatives. And in fact, to assess the biomarker clinical value, we need to have biomarker positive and biomarker negative patients in a trial. Of course, we may have other source of evidence, preclinical evidence, that BRAF and wild type patients cannot respond to dabrafenib. And okay, but we can't estimate the clinical utility of the biomarker. So we could also try to test for interactions, and I have here two examples, C and D, where there are interactions between the biomarker and the treatment. Actually, on a logit scale, the level of interaction is the same on the left, on the right. But the conclusions are not the same. For the right marker, the D, we have already seen that. The DFS rate is always better with chemotherapy than without chemotherapy. So even when there is an interaction, it's not, in that case, it's not useful. On the left, we have some patients for whom the plain curve is above the dashed one, and some patients for whom the dashed curve is above the plain one. And here is a useful marker for treatment selection, because in some cases, for some values of the biomarker, you have a better survival with one treatment, and for others, you have a better survival with or DFS for the other ones. So the marker becomes really useful on the left. The interaction alone is not sufficient. So what Holly James and colleagues have proposed is to draw marker by treatment predictiveness curves. So here on the right, I have put a similar curve that I have done myself. And then when the curves cross, we may define these as the line that separates marker negatives on the left and marker positives on the right, positives being the ones for whom chemotherapy gives a better DFS rate than tamoxifen alone. And we can define for any patients with a value X of the marker, a value E of X, which is the difference of the probability of a good outcome for those when they're receiving the treatment, Z equal 1, and those not receiving the treatment, Z equals 0. And if E of X is positive, then we define the patients have a marker positive, and we say, well, those should be treated by the new treatment Z1. The other can keep Z equals 0. And if we do that, we get the red curve for the overall survival curve. And you see we get the maximum that we can obtain, which is when they are negatives, they receive no chemotherapy and tamoxifen alone is better. And when they are positive, they receive tamoxifen plus chemotherapy, which does better than tamoxifen alone. How can we measure the performance of that marker? We can measure it as the change in outcome under a marker-based treatment strategy versus a non-marker-based treatment strategy. It depends on the value E of X, but it also depends on the proportion of the population that will be affected by the marker and the change of treatment when we measure the biomarker. In our example, tamoxifen plus chemotherapy does better than tamoxifen alone, so we will start from this one as a reference strategy or the standard strategy. So what we obtain in average, the 79% 5-year DFS, is the probability of Y equal 1 of a good outcome given Z equal 1 because we give chemotherapy Z1 to everyone. Under the marker-based strategy, we only give chemotherapy to those who have E of X positive. So the expected outcome under the marker-based strategy is the probability. I try to, anytime I touch something, it moves. I try to take that. Sorry. So this one only for the marker positives, and this is multiplied by the proportion of whom receive chemotherapy. And the other one have the outcome they have under tamoxifen alone. And we can compute the difference of both. And the difference of both gives us the mean of minus E of X when E of X is negative, so actually this is positive, multiplied by the proportion of marker negatives. Sorry, I have to. So if we come back to our markers, we had marker C who seemed useful, and if we use the marker-based treatment strategy, we will obtain 80% overall DFS instead of 79% when everyone received chemotherapy. So it's not a huge increase, but we can spare chemotherapy to 44% of women, and this could also spare the adverse events of chemotherapy. So it could be beneficial on other aspects, and it increases a bit the overall DFS rate. With the marker D, we have no marker negative, everyone is marker positive, so we get, if this one leads to giving chemotherapy to everyone, so it doesn't change the overall DFS. And marker E is a bit like marker C, but much more pronounced. So if we have such a marker, we can increase a lot the DFS of the entire cohort of women by giving the right treatment to everyone, and by also changing treatment for only 45% of women. What I'm talking about was for a continuous marker, and I would agree that many markers are binary, but the evaluation is even simpler. We don't have to do the curves and to define marker positivity. We can just compare the strategy without the marker and the strategy with the marker. We should still be careful that many binary markers are not binary at the beginning, but are dichotomized at a later stage. So perhaps it's better to draw the curve and to find the right threshold. But in any case, we can do that. Thanks for your attention, and I'm happy if you have questions on the biomarkers.
Thank you, Christine and Raphaël, for your excellent presentations. We will now address questions from the audience. Please feel free to add those to the question box. So the first question is for Christine. Could you please tell us how to get involved in ECRIN Data Center certification program?
Yes, thank you for this question, Laurent. So there is, as I said, a kind of procedure for application, which is for the applicant to fill in a questionnaire. This questionnaire is really used by us to assess the readiness for an audit. And for a certification audit, and for member countries and observer countries of ECRIN in Europe. This goes also through the direct contact with their network. So in practice, 2 contacts to be taken. One first contact directly via the link that is on the ECRIN website, which is coming to ECRIN directly, and another which is at national level with the national network that you are part of.
Okay, thank you. A second question to you, then we will switch to Raphaël. What recommendations do you have for preclinical researchers to maximize success in translation to clinical stage development?
Good question, and quite complex to answer in just a few minutes. Preclinical research is really already arbitrated, I would say, and you have very good requirements that are detailed and set up in the consensus GLP documents that is dedicated to computerized systems and data management in non-clinical research. So this is one first direction that could be read, and you can take knowledge of this. And the other point is really to so I can provide you with the reference for this document, and then quite often the issue is to make the link in between the preclinical research and the clinical research. And here you have a number of documents and guidelines related to first in human that could be interested to read. And I hope that I answered this question. If it's not the case, I can give offline more information about this.
Thank you very much. Now we have a question for Raphaël. If you would like to engage in biomarker utility, what recent publications would you suggest?
It's not an easy topic. Clearly, reading the 2011, that's a recent paper that I've used here, is good. There are later papers, so I can suggest other papers by Holly James in particular, but they are quite more statistical, so it really depends on the level of statistical proficiency. Basically, it doesn't change so much as compared to that more light vision, but they have made some refinements. And then there is so it's for a single biomarker. Now there is also a trend to going to individualize treatment effects, which tries to predict using regression model or machine learning algorithms the outcome that someone would have under a specific treatment and under another treatment control, and to compute the difference, and then to have some individualized treatment effects. And there is a new guide for those techniques, which is called PATH, a reporting guideline. PATH, it has been published, it's perhaps still ahead of print, but it dates back to November 2019 in Annals of Internal Medicine. So if you want to go to those, either you are very much interested in biomarkers, so you can read the James paper, or you are more interested in predicting individualized treatment effects, which is now at the core of precision medicine, I think. And then you could go to path.
Okay, thank you very much. A last question for Raphaël. Should we always ask for clinical trials when investigating clinical utility?
That's a very, very clever but difficult question. If we look at those papers, the guidance, and it was for a long time my view that yes, otherwise you don't have the clinical utility. So sometimes you may have, I would say, preclinical evidence that it's useless to do so. For instance, BRAF inhibitors are more than unlikely to work if the tumor is not BRAF mutated. Though all may not be so specific of BRAF mutations and could, and there are other RAS pathway mutation. So we have to have very, very strong preclinical or pharmacological evidence not to go for a randomized trial, for an all-comer trial. So a trial where you have biomarker positives and biomarker negatives. And that's really, I think, the gold standard to evaluate the clinical utility of a marker. Now, if the question was about randomization, I clearly believe that we should go for randomization, and that single-arm trials are not suitable for for precision medicine, and enriched single-arm trials are the worst we can do for that. So it's okay to see whether the drug has some activity, even very nice activity in a specific subpopulation, but it does not answer the question whether the PFS or survival is better than control, neither whether the biomarker negatives cannot benefit somehow from the drug. So yeah, I'm still very much in favor of randomization here.
Thank you, Raphaël. We have now come to the end of this webinar. I would like to thank you all for attending today's webinar, with a special thanks to our lecturers for their excellent presentations. Should you still have any further questions to one of the speakers, I welcome you to submit those to us by email, and we will get back to you as soon as possible. The recording of this webinar will be made available to anyone interested on our website within 2 weeks. We hope to see you all again soon. Thank you and have a nice day. Bye-bye.